Hello everyone, this is the third chapter, cell, its structure and functions, and all components of cell and their diagrams. So cell is the fundamental unit of life. A cell consists of different components which help the cell to run properly. Each of them has a unique set of functions. Our body contains different type of cell like muscle cells, nerve cells, etc. having different structures. So all of them have similar basic components. In the following notes on biology, we will learn about the various components of cells and their functions. So cell membrane, this is also known as plasma membrane, and this is made up of protein and lipids. There are so many models given by various scientists, but the fluid mosaic model was proposed by Singer and Nicholson in 1972. This model describes the structure of plasma membrane as a mosaic components including phospholipids, proteins, cholesterol and carbohydrates that give the membrane a fluid character. Example, characteristic shape of human blood cells, bone cells and nerve cells. It allows the transport of selective substances into and out of the cell, but not all substances. This is why it is known as selectively permeable. Because you know membrane is of three types: impermeable membrane, second one is selectively permeable, third one is permeable. So functions of plasma membrane. The plasma membrane encloses the entire cell contains. It provides the cell shape. Plasma membrane help in the transport of material in and out of the cell. Small molecules can be transported across the plasma membrane by any one of the following three methods. Diffusion. In diffusion, molecules of substances move from the region of higher concentration to the region of lower concentration. Diffusion does not require energy. Example, absorption of glucose in a cell. And if you kept a perfume bottle in a corner, this perfume diffuse in all over the room. So, and second one is osmosis. It is a process of movement of water molecules from the region of the higher concentration to the region of their lower concentration through a membrane. Next is active transport. When the direction of movement of a certain molecule is from the region of their lower concentration towards the region of their higher concentration, it would require an active effort by the cell, for which energy is needed. This energy is provided by ATP. The active transport may occur through a carrier molecules like sodium-potassium pump. Next one is cell wall. This is the outermost layer and it is composed of cellulose and found in plant cell. It protects inner part of the cell. It is rigid that gives shape to the cell and does not allow distension of the cell, thus leading to the turgidity of the cell that is useful. Next function. So it freely allows the movement of water and other chemicals into and out of the cell. There are breaks in the primary wall of the adjacent cells through which the cytoplasm of one cell is connected with the other. These cytoplasmic strands which connect one cell to the other one are termed plasmodesmata. Plasmodesmata is connected to cell wall, is found between two cell wall. Endoplasmic reticulum, short form is ER. It was discovered by Porter in 1954. It is absent in human RBC, BGA and bacteria. and it helps production and storage and transportation of molecules. ER is of two types, rough and smooth, SER and RER. And जो that is RER, rough endoplasmic reticulum. Ribosomes are present on its surface. It is involved in protein assembly where it synthesizes and packages important proteins. And smooth ER, endoplasmic reticulum, here no ribosomes are present and its function is to synthesize lipids and steroids and the storage of carbohydrates. This is the structure of ER. You can see smooth and rough endoplasmic reticulum outside the nucleus. Ribosomes are present in the cytoplasm of a cell and also found on the surface of ER. They are made up of RNA and proteins. Functions: nicknamed protein factory of the cell. This organelle is responsible for the synthesis of protein molecules. It collects important amino acids and then produces protein chains based on what the cell needs. Next one is Golgi apparatus. It was discovered in 1898 by Camillo Golgi. There are 3 to 7 Golgi bodies present in an animal cell but not present in BGA, blue-green alga. So Golgi apparatus in plant is also called as dictyosomes. Many of these organelles are present in a plant cell. In animal cell, the Golgi apparatus is present near the nucleus. In plant cell, they are present all over and around the cell. This is the structure of Golgi apparatus. Function: its function is to store and transport protein and lipids that have been synthesized. It is also help with the synthesis and secretion of important molecules. It helps in synthesize wall element like pectin, mucilage, and proteins are stored in a temporary vacuole. This is called lipochondria. Lysosomes are also known as suicide bags because lysosomes secrete some digestive enzymes that dissolve all organelles of the cell. It was discovered in 1955 by Christian D. Dewey. Lysosomes are pouch-like structures found only in animal cells. They are special vesicles that are pinched out of the Golgi bodies. They contain different digestive enzymes like protease, phosphatase, glycosidase, and sulfatase. They are called suicidal bags of cells because they contain lytic enzymes. With these enzymes, lysosomes digest their own cell, causing the cell to die. This mechanism is used when the cell is old or diseased. Function is lysosomes digest nucleic acid, polysaccharides, fats, and proteins. The main function of lysosome is intracellular digestion. Function like digesting food, breaking down a dying cell, or digestion of unwanted substances. So this process is called autolysis. Lysosomes can end up killing themselves. Next is plastids. These are found in plant cell. The term plastid was first coined by Ernest Haeckel in 1866. These are absent in animals, BGA and bacteria. 3 types of plastids are found. Leucoplast, these are colorless. Chromoplast, these are colored. And chloroplast, these are green in color. This is the complete structure: etioplast, chromoplast, chloroplast, leucoplast mainly found in roots, amyloplast, elioplast, and pretinoplast. Next one is cytoplasm. cytoplasm is a fluid which is found between nuclear membrane and plasma membrane. It is present inside the cell. It is present only within the cell membrane of a cell. It is not present in the nucleus. Another fluid is present in nucleus that is called nucleoplasm. So it comprises mainly water but also enzymes, salts, organelles and various organic molecules. Function is all the cell organelles such as nucleus, ribosomes, mitochondria, Golgi bodies, plastids, lysosomes and endoplasmic reticulum are present here. Cytoplasm support them and keep them nourished. Mitochondria, these are also called powerhouse of the cell because they produced ATP, adenosine triphosphate. They are present in both plants and animals. Mitochondria in animal and plant cell are the energy generators and releasers. Mitochondria appear as tiny thread-like structure under the light microscope. Size is 0.5 to 1 micrometer. In structure, mitochondria is double-walled structure. The wall is made up of double membrane but is categorized into 5 distinct compartments. The outer mitochondrial membrane, inner membrane space is the space between the outer and inner membrane. Inner mitochondrial membrane, the cristae space formed by infolding of the inner membrane, and the matrix is space within the inner membrane. This is the structure of mitochondria. You can see inner membrane, here finger-like inner growth in the matrix. These finger-like growth, these are called cristae, and F1 particles are found on the cristae. Function, mitochondria convert glucose into ATP through 5 processes: glycolysis, CTA and ATP synthesis. Through the process of cellular respiration, mitochondria oxidizes pyruvate in its matrix to form pyruvic acid in order to capture the energy that is stored in the form of ATP. This is why mitochondria is called powerhouse of a cell. This ATP is then digested by the organelle in a cell to produce energy for their functioning. ATP digestive enzymes. Nucleus, this is the important organelle of the cell. Robert Brown was responsible for discovering the nucleus. He discovered it in 1831. It stains deeply, spherical in shape. WBC has lobed nuclei. Nucleus and chromosomes are found in the nucleus, and chromosomes are stained by acetylcarmine. Mostly there is only one present in each cell. This is a double-layered structure having fine nuclear pore encloses nucleoplasm, which contain a chromatin network and a nucleolus. Component, a double-layered membrane called the nuclear envelope, which has a large number of nuclear pores. This membrane is made up of lipids and proteins like plasma membrane and has ribosomes attached to the outer membrane, which make the outer membrane rough. The pores allows the transport of large molecules in and out of the nucleus, and the membrane keep the hereditary material in contact with the rest of the cell. Nucleoplasm, liquid found within the nucleus. Just like the cytoplasm is found within the cell membrane, like this nucleoplasm is found within the nuclear membrane. It supports and surrounds all components of the nucleus like DNA, RNA, chromosomes and nucleoli. Chromatin, within the nuclear membrane, there is a jelly-like substance known as karyolymph or nucleoplasm that is rich in proteins. It is made up of histone and nucleic acid. The fibrillar structure forms a network called chromatin fibrils, which gets condensed to form distinct bodies called chromosomes during cell division. The point-like structure is called a gene. On staining the chromosomes, two regions can be identified in the chromatin material: heterochromatin dark and euchromatin light. Heterochromatin has highly coiled DNA and is genetically less active than euchromatin, which has highly uncoiled DNA and more active genetically. This is the structure of chromatin and condensed chromosome structure. You can see DNA helix also. The nucleolus. Felix Fontana discovered the nucleolus in 1774. It is a suborganelle found within the nucleus of a cell. The membrane has fewer spheroidal bodies present in all eukaryotic cells except in sperms and in some algae. It is composed of DNA, RNA and proteins. Around 85% proteins, RNA, 10% and DNA 5%. Storehouse for RNA and proteins, it briefly disappears during the early phase of the cell cycle and reappears after telophase in the newly formed daughter nuclei. You can see what is the telophase and cell cycle in cell division. Regulate the synthetic activity of the nucleus. The nucleus and cytoplasm are interdependent, and this process is equal to nucleocytoplasmic interaction. These are the questions. You have to write down answer of all these questions. Who discovered the Golgi apparatus? Who discovered nucleus? Who discovered plastids? Endoplasmic reticulum. Why is cell membrane called selectively permeable? Who discovered mitochondria? Thanks for watching. We will meet again tomorrow in our next lecture.